So just going to make a few introductory points here. I wanted to mention that we are here in large part because of Dr. Javid Bigwell and his lab. We are going to be talking about things related to RNA-Seq, but in the context of those things that were pointed out by him and his collaborators that are important right now. The presenters are going to be Dr. Vladimir Galatenko. He's the chief scientist at the Tauber Bioinformatics Research Center, and he's going to be presenting together with Julia Panoff. She's a PhD student, and both of them will share both biological interpretation or biological projects and some technical things. This work is supported by the Tauber Bioinformatics Research Center, which is at University of Haifa in Israel, and also Pine Biotech. When we are going to be talking about some data, examples based on data, these are published data sets. We provide here links to those published data sets. You're welcome to read the publications and also download this data. It's available. And also I want to point out that some of these projects are prepared as educational projects and are available on our platform. Our platform is the, first of all, the website that you're on right now, edu.t-bio.info. And you will find there are also some courses on basics of data analysis, similar to what you're going to be seeing here today. So just an overview of what we will cover today, or hope to cover. First of all, it's going to be about next generation sequencing data pre-processing, including trimming technical sequences and removing PCR duplicates. And the second point about processing of next generation sequencing data is going to be around quantification of expression levels, conventional pipelines, and also identification of novel isoforms. We will touch on the challenge of gene set enrichment analysis, so annotation of genes, but also gene set enrichment. And finally, we will practice and utilize some machine learning methods, including unsupervised analysis of expression data and supervised analysis. So now I'm going to hand over the presentation to Julia. And Julia will share with you her presentation about the biological examples. Okay. So Julia, you should be able to share your screen now.
Okay, hi. Can you see my screen? Sure. Can you see my screen?
Yes.
Okay, great. So, hi everyone. As Ilya mentioned, my name is Julia, and I use the platform in my research for neurodevelopmental disorders. And I also collaborate and assist other groups in using the platform in their research in areas such as marine biology, evolutionary biology, and cancer studies. Wait, but I'm actually on the wrong slide, right? Okay.
Yeah. You want me to show your slides? Okay, Julia, I have your slides here if you want to continue.
Okay, I'm trying to get this working. I'm sorry for some. Can you see the screen? Hopefully. Sorry. So, okay, so my goal today, before we begin the hands-on part of the workshop, is to give you a taste of what we can do with the Tauber Bioinformatics platform, and also with bioinformatics in general. So I will focus on one project that we have been working on, and I will briefly tell you about the kind of data and the analysis we used to generate the results that I will present afterwards. Every step of the analysis that you will be hearing about was done on the platform. So let me begin. Yeah, I'm on the wrong slide. I'm not in the present mode. Как мне в другую сторону? Wait, just a minute. Ilya?
Yes.
Sorry about this. My sharing screen is not working for me. Could you do it for me and share the presentation for me, and I will just speak?
Yes. Okay. Okay. Can you hear or can you see my screen?
Yeah. Okay, great. So, so maybe you can show the next slide. So I will focus on one project about cell lines, and I will tell you about the kind of data and the analysis that we use to generate the results that I present afterwards. So let me begin. The project we will be looking at is based on the analysis by the Gray Group, and I hope you had a chance to look at the publication, but if you didn't, it's okay. We will be, I will go over that as well. So the original data consists of 70 cell lines of breast cancer, and we used only 49 of these cell lines that had all the molecular profiles for them. As you can see on the slide, we had gene expression data and RNA editing data. So the RNA-Seq data is actually produced by sequencing the RNA molecules in the sample. So from this RNA sequencing, we can get two kinds of data. We can get the gene expression data that we will go into more detail, and you will have a chance yourself to practice how to do it exactly. And also we could do RNA editing. We could generate the RNA editing table from this data as well. So what is editing? RNA editing is, for those of you who might have forgotten or don't know, is a post-transcriptional modification of primary RNA molecules. as you can see on the slide, sometimes RNA molecules are edited by a special enzyme, and there are different types of editing. There is a substitution of cytosine to uracil that in the sequencing data we see as a thymine. And another type of modification could be substitution of adenine by guanine. So we studied these two modifications together with gene expression. And RNA editing events can have very big effects on afterwards on translated protein. The protein can be unfunctional, or it can lose the binding sites for microRNAs that it had in wild type state. The exact mechanism and the function of editing events are not completely understood, but it has been shown that RNA editing plays a significant role in different types of cancers and other disorders. So this is about the kind of data we were looking at, gene expression and RNA editing. And the question is why? Why would someone might be interested in data-driven analysis that actually has almost no planning beforehand? We were not looking at specific genes or perturbated states. We were just looking at the data overall. So we were interested actually in trying to identify subtypes of breast cancer. As you know, breast cancer is a heterogeneous disease, and our understanding of its complexity is still evolving. So on this slide, you can see kind of our history of what it was, how people have, with time, have been trying to identify the subtypes of cancer, breast cancer. And recently, people are trying to identify subtypes of cancer based on what we call multi-omics analysis. We combine different layers of omics, RNA data, DNA data, ChIP-seq data, all together in one analysis to find specific unique molecular characteristics of subtypes of cancer. And as you probably know, one of the methods or models of cancer are cell lines. And these cell lines are subtypes of cancer or types of cancer. And they are well-established cell lines, originally taken from patients that underwent biopsies. And they are very useful models for studying the cancer. As you can see on the next slide, we can have profiles of different types of cell lines with genomic profile as well as pharmacological profile. And based on these profiles, we can better find treatments for patients that have similar genomic profile. So, of course, cell lines are not the best models of cancer. They lack some of the important things that cancer in patients have, and that is heterogeneity inside the tumor cells, also the stromal component. However, despite the limitations, cell lines continue to be very important research models and preclinical trial tools in cancer biology. So understanding the characteristics of different cell lines can help us better understand both the subtypes of cancer and also sometimes understand that some cancer line is not a good model for the type of cancer that it was used for as a model. So in on the next slide, you will see that our project was based on 49 such cell lines. We had gene expression table, and you can see on the slide, the gene expression table is actually a collection of all genes in the sample You have different samples with different profiles for each gene, so expression levels. And on the bottom table, RNA editing table, you see chromosomal regions on the genome that have RNA editing events with high probability. What we were trying to do, on the next slide, you will see that we need to take these two tables and find groups of genes that are, and groups of RNA editing that together have the similar sample profiles. So you can see on the slide that, for example, we could find a group of genes in yellow and a group of RNA editing events also in yellow. And they would give us clustering of samples that are very similar to each other. You can see the purple arrows pointing to the clustering of the samples. And those genes and RNA editing events would be somehow linked. Or another case could be that the genes in green and RNA editing events in green, that could also give us a separation of samples, a different separation of samples. Or, for example, an outlier sample that is somehow unique in its molecular characteristics. So the goal of our analysis in this project was exactly that: to find these somehow different samples that are different in molecular characteristics from all others. So this is actually what we were able to do, was to identify one unique cell line that was very different from all others. On the next slide, you will see matrices of distances between samples. So those are distances between samples. And you can see in blue, there are lines in blue on both tables. One table is distances of samples based on the gene expression table. And on the other side is the matrix of distances between samples based on the RNA editing events. And you can see that both of these tables actually separate only one cell line, HCC 202. You can see that separation in blue. The distances between HCC 202 and all other cell lines are very high, and they are painted in blue for visual representation. So another way of showing this is going to be on the next slide, is to look at the PCA of the actual, if you took now these genes and you would look at the PCA based on the expression level of these genes, you would see that these genes that we chose are only present in this sample. And also with RNA editing events, they are also only present in this sample. So without actually designing our experiment so that we could target specific genes, we were still able to choose a group of genes and a group of RNA editing events that clearly separates this sample from all others. So let's look a little bit at what kind of genes these are. On one side, we found olfactory receptors and microRNAs. Those were the expression levels. And on the other side, we found Rab GTPases. So can we, from knowing these genes, can we now say anything about this cell line? How is it different from all others? Well, we could have some hypotheses, and afterwards we could maybe generate more experiments or take more data to validate our hypothesis. One of the hypotheses we were thinking of is that microRNAs are known to bind to genes and to prevent the expression of these genes. And microRNAs are also known to bind to Rab GTPases, which are very important transport molecules. And when RAB GTPases are edited, sometimes these binding sites are missing, and no longer microRNAs can bind to these genes. Because of that, we would have a very high expression of transport molecules, RAB GTPases. And it's known in cancer studies that olfactory receptors can cause malignancy in cancer or aggressiveness in cancer. So RAB GTPases, by trafficking the olfactory receptors to nearby cells, can actually make the cancer cell line or the cancer tumor much more aggressive. So this would be some kind of a hypothesis that could be validated more with more targeted experiments later on. So this was a brief glimpse at what can be done with data-driven bioinformatics approach. And we hope that now you have a little bit of a more understanding of why this approach can be helpful in biological studies and life sciences. And now I hope you will enjoy the hands-on workshop. So thank you very much for your attention.
Okay, thank you, Julia. Now I'm going to switch over to Vladimir. As it was said before, Vladimir is the chief scientist at the Tauber Bioinformatics Research Center. He is working with the team at that research center on development of the platform and extending it for both research and educational purposes.
А можно микрофон, чтобы динамик вырубить? И микрофон, и звук. Ага. Okay. Okay, so hope that the screen is shared correctly. If there are any problem, please let me know. And before we start, please let us check that all of you have an access to your accounts at the Tauber Bioinfo platform and that you have all the files in the archive prepared for this workshop. So please let us know if anyone does not have these files, as we'll use them later on in our practice. So in any case, we begin with some theory and then we'll proceed to practical analysis. Hopefully the theory will be relatively short and it will be related to the basis of RNA sequencing pipeline. I mean, library preparation, the steps that we need to understand in order to understand what are the main steps of the analysis. As you know perfectly well, everything starts with the genome, which part codes proteins, so genes code for proteins, but proteins are not generated just directly from genome. There's an intermediate step. Genes give rise to transcripts, to messenger RNAs, and these messenger RNAs are then translated to proteins. And all the cells for the fixed organism, for example, all the cells for one human, well, generally, if we avoid thinking about cancer cells for a while, generally have the same genes, but these genes have different activity, different levels of activity, different expression levels in different cells, in different tissues. So some genes may be present in the genome, but give rise to no messenger RNAs, no transcripts, and hence no proteins. Some can give rise to an essential amount of transcripts and thus essential amount of proteins. And roughly speaking, sequencing process can be described as follows. We have all these transcripts. These transcripts are randomly divided into small pieces, and these pieces are read by a sequencing machine. So read letter by letter, nucleotide by nucleotide. The problem is that we do not know the origin for all these pieces we get after this reading. But anyway, we know the sequence for human genome, we know nucleotide sequences for all the genes, so we can map our short segments on transcripts, or better to say, probably better to say, on genes. And if there are no reads mapped on a gene, for example, gene B, we see that this gene has 0 or at least low expression level. More reads, more short segments we have, the higher the expression level is. But if we go deeper in the details, we'll see that this process has several intermediate steps. These steps might be slightly different between technologies, but generally these steps are the following. First, all the transcripts are shattered into subpieces. So we have shattering, and in fact we have one copy of each segment. More copies of the transcript of a transcript we have, more pieces related to this gene we observed in our sample after shattering. At the next step, adapters are ligated to these small pieces, in fact to these pre-reads. In fact, this ligation is successful not for all small pieces, but at least these adapters are ligated to a sufficient part of these short reads. Then PCR amplification is performed, that is, multiple copies of each read with ligated adapters is generated. For example, if there are 6 rounds of PCR amplification, well, if there are 2 rounds of PCR amplification, we get 2 multiplied by 2, 4 copies for each piece. If we have 6 rounds of PCR amplification, we get 2 power 6, so 64 copies. More rounds we have, we get more copies for each segment. There's a slight problem related to the fact that PCR efficiency is different for different segments. So for some pieces, we have more copies. For some pieces, we have lower numbers of copies. So we're speaking about average numbers of copies we obtain at this step. So please pay your attention to the fact that indeed there are multiple copies of each segment after this step. But sequencing takes not all the pieces, but takes only a relatively small fraction of these pieces and reads this small fraction. If the number of copies is relatively low, it's the case when the initial amount of RNA is sufficient and the quality of library preparation is also sufficient. Then generally we'll read only 1 copy for each short segment. Number of duplicates, so number of reads originated from the same small piece of initial transcript, will be well below 10%. If we start with lower amount of RNA, so we need to perform more steps, more cycles of PCR amplification, we get more copies for each segment, and the number of these PCR duplicates increases. Moreover, the absence of uniformity in the number of copies starts to play the role. But after that, we again continue to our previous discussion. We simply map all these reads on initial genes and estimate an expression level just counting reads mapped on each gene. So this logic explains us the major steps of the analysis. We may start with preprocessing. So if there are additional segments ligated, additional sequences ligated to our initial segments adapters, these adapters may be removed prior to the analysis. Additionally, there's a standard fact of low quality ends for reads. They also can be trimmed to increase mapping quality. Then PCR duplicates, so identical reads that are potentially the copies of the same initial segment, can be removed from the analysis. These steps increase the quality of the resultant mapping. So we map reads on known genes, on known transcripts. But at this stage, there are different strategies. We may focus just on estimation on expression level of known genes. In this case, we map all these reads to the set of known genes, known transcripts, known isoforms. We can also want to try to identify novel transcripts, novel isoforms of known genes, or completely novel transcripts. It's a more rare type of the analysis, but it's also not technically hard. And definitely there's a combined strategy. After the reads are mapped, we can proceed to the quantification of expression level. So just in order to understand how it can be performed technically without going deep into the details of mapping algorithms, of the estimation algorithms, let us get started with the use of the Tauber Bioinformatics platform. Let us prepare the pipeline that performs all these jobs we've discussed. We won't run this pipeline as the size of sequencing data is generally relatively large, so processing time is also relatively large. It may take hours or even several days if we have many samples. So we'll just prepare this pipeline. In order to do it, we proceed to the platform. Please let us know if there are any problem with it. And we go to the analysis related to RNA sequencing data. So here it is. So yeah, there's short introduction. So what we can choose here? What are the options? Today we're dealing with human genome. So we select the set of genes and the genome as you see. So we select Homo sapiens. There are two types of reads, single end and pair end. For example, for our today discussion, we have some pair end data, and we need to okay. Perfect. Yeah. I think I see it from here. Great, thank you. So let's just try to do it. Okay, and the size of the size of sequencing data is large, so uploading of new files can also be slow. That's why we use just links to the files that are already inside the system to have this short test. So,
sorry, Vladimir, somebody was just asking if I think just to make sure, Sahil, is everybody on the same page? Can we move forward, or is somebody having issues?
Yeah, just give me a minute. Go back one slide.
Okay, if you go back one slide, Libby.
Okay, currently I'm just sharing screen, so it's again we are inside the platform. So when we log in, we can press on the areas of the analysis link, and then we've got sections for analysis of various types of omics data and for various preprocessing. Here we go to the analysis of RNA sequencing data. So here's the link on the top, left top link. So here we are. Here, in order to perform the analysis, to perform standard analysis, we use type of sequencing data. We have single-end sequencing or pair-end sequencing. Well, currently, in many cases, it's pair-end, PE, pair-end, and select genome or the set of genes, select the transcriptome where working is. So pair-end and Homo sapiens. And then we upload files. So we press on upload files and add groups button. Please let me know if there are any problem with what's going on. And for our case, for the case when we use files that are already inside, we use URL bulk upload. Here it is. So here we go to the files in the archive you have, main files. And for example, if we want to start with just 2 samples, we use the file links to samples.svl. So this is a very small file which contains only links to our data, but not the data itself. Okay, so dear colleagues, if you do not have these files, please let us know and we'll simply fix this problem.
There was a question. So what are the 4 drop-down options?
So, okay. So first, we can deal with different types of experimental data. I mean, model organisms for which genome or transcriptome is known. So, and we have everything known for an organism like Homo sapiens. So it's the first option. Data type we're dealing is, so typically for RNA sequencing, we start with FASTQ files. If we begin from the very beginning, then the type of reads, single end or pair end. And if we're dealing with a model organism, we just select genomic and transcriptomic data for related to this organism. So organism and reads type. So then, okay, then we upload a file from our set.
Okay, just to clarify about our set, what we mean is there is a button to download files on the page that you're on. So you should have downloaded the zip file, and that zip file contains multiple folders. One of the folders is called
So main files, where we'll mainly deal with main files.
So you download the file, you unzip the file, inside you see the folder called main files, and then we will be using the files that have an extension .svl.
So, okay, and we select this file. Yes, hopefully we'll get used to it promptly, as we'll create a number of pipelines today. So the file is links to samples. And then we go further with our analysis. Here, we're not going to make differential expression analysis or any complicated analysis. Currently, we're focused on just estimation of expression level. So after pressing define groups, we simply say that we need no groups, no additional information for this analysis. So cancel no groups. And then we start creating our pipeline. So the pipeline should be started. So again, I select start. Then a possible option is okay. Here again, so let me just show it once again. Here we use bulk upload, as we're uploading only links to files, but not the files themselves. So later on, we'll use main uploading button, add files, but currently we're using bulk upload button. And upload the file links to samples.svl. So we then press on define groups and say that we have no groups and go on. So, okay, let's take a look whether there are any problems at this stage. If not, we'll try to go further. Okay. So we initialize our pipeline with the start module, start and OK. Then for trimming adapters and low quality ends of reads, we can, for example, use Trimmomatic module. We press on the module and then confirm by pressing save. PCR duplicate removal can be performed via the module PCR clean. So default technical option is perfectly okay. Mapping can be performed if we're going to map all our reads on genes on transcriptome. For example, with bow tie or BWA, for example, with bow tie. And expression level quantification can be performed via RSM expression table module. Here, we need to choose the units for expression level quantification. We'll discuss these units in a minute or two. So, for example, fragments per kilobase per million map reads, FPKM, we'll also do. And that's all. So this model will give us quantifications for expression levels. Here, we can just finish our pipeline, end model, and save. Give a name to this pipeline, for example, test pipeline, or whatever you like. And then, in order to run pipeline, we won't do it now, as this pipeline takes okay. So now we won't run this pipeline. We just looked at how this pipeline can be created. So while we're trying to solve our problem, let me just repeat all these steps
Yeah,
in order.
Vladimir, some people are saying that they are still making the pipeline. So yeah, if you can,
Okay,
that would be
great. Please let me know when everything is okay. I'll just repeat all the steps for people who are going on, just to make visual what we're doing. So I just recreate this pipeline. I start with the areas of the analysis, RNA sequencing, select parent sequencing, select genome, upload our file links to samples, tell that I need no groups for the analysis, then start, for example, trim adapters. If I select a model which is not required, I select a trimmatic, but I want to remove it from the pipeline, I can press just right mouse button and remove it. So removing is performed via the right mouse button. But I add trimmatic, I add PCR cleaning, I use mapping, this time, for example, bow tie. After mapping, I need to go from map reads from read positions to expression level quantification, which is performed by RSM expression table. And that's all. And that is all. And pipeline name. And in order to start this pipeline, I need to press run pipeline, but currently I won't do it. Okay, so is everything okay with this simple test? Okay. So anyway, it's just an example, and as you see, we can easily include different types of modules for each step in our analysis. For example, I'm just going on with my audio because it's not essential things. I just give several comments for people who have already created these pipeline. So there are different modes. For example, adding mapping on genome and cuff-links, cuff-merge models will also allow us to add steps related to the identification of novel isoforms. So algorithmically, it's not so simple. But as we have all these modules, all these algorithms inside the platform, the usage is really simple. We add additional modules to the pipeline and wait for the result. More modules, more execution time, but definitely it's not a problem for the majority of experiments when the data is collected for weeks, months, and sometimes even for years. So additional data of the analysis is sometimes not essential, but from the other hand, faster analysis is definitely what we also want to have.
Vladimir.
Yeah.
So there are some questions that Sahil wants to ask on behalf of some people. So if you have some questions, let's just go through them right now.
So please feel free to ask at any time.
There are several buttons, right? You select PCR clean, then you go to mapping transcript. We don't know, we couldn't follow that. Because first you can explain all the things, what is error correction, what is exon detection, then mapping transcript. If you explain those things, then if you explain the schematic, then that will be helpful. You just ask us to click this one means we don't know.
Okay, so what I understood is that the question is, what do these buttons actually do? To explain a little bit more in detail what Trimomatic and PCR Clean do, so that it's clear to everyone it's not just buttons on the screen.
Okay, so thank you very much for your question, and let us return to our standard scheme of the analysis of RNA sequencing, which just follow our general idea of the preparation of experimental data. So first, we ligated the adapters to our short sequences, and standard pipeline may want to remove these short sequences. This is performed by the first model we added, Trimomatic. So Trimomatic looks at the ends of reads, tries to identify these standard adapters, and this model also has functionality to try to identify novel adapters which are not in its database, and performs just trimming. So removes these adapters from the reads we have sequenced. Then we discussed PCR duplicates. And so our next step was to remove these PCR duplicates. And this is what is done by the PCR clean algorithm. So it just looks for pairs, triples, quadruples, and so on of identical reads and leaves only one copy. So performs these PCR duplicate removal. The next step is mapping these short reads on genes. So for each gene and for each short segment, we want to find out whether this read has a good position above this gene. So, and after this stage, we've got positions on the transcriptome set for each read. Some reads can have no allowed positions, and for some reads, we may fail to identify its origin. Some reads can be mapped on different locations, but all this information is obtained by the mapping model. For example, as we're mapping on the transcriptomic data, bow tie, BWA, something like that. And the final step is going from read counts to go from this mapping to the identification of expression levels, to the quantification of expression levels. And it's performed by the RSM expression table module. So definitely algorithms that lie behind these titles are not very simple, especially if we're speaking about mapping and quantification, but the purpose of these models is just as I described. So let us please now have a very short pause, I suppose for 3 or 4 minutes, just a short technical break. And after this break, we'll try to discuss what are the standard units of quantification, why we should go from raw read counts to some more complex measures. So currently, just for a moment, just for several minutes.
Okay, can everyone hear me? Okay, during this break, it would be helpful, I assume that everybody had a chance to take a look at the first pipeline that we created. Let me turn on my video here. Okay. Okay, so while we're taking this short little break, I'm going to turn on the microphone for Sahil. And please, Sahil, if you want to just make sure that anyone has questions.
In the action in your platform, the quality control of Q files. The system data will make shows the first Q file.
Sorry, I could not hear that. Sahil, can you just repeat maybe here next to the computer?
Hello. Hello. Is there any option for the quality control of HQ file before processing of the data?
Yeah. So we, you have the quality of reads, and I think we can touch on that subject. But in the meantime, what I want to do in this short break is just maybe collect all of the questions, and so we will be able to answer them one by one. So we noted it down. Question number 1 is quality of raw FASTQ files before you start the mapping.
Thank you.
Is there any way to get the details regarding each and every step?
Okay.
How to identify isoforms using this pipeline?
Okay, about the isoforms, just a quick note, we will get to that. Go ahead. Are there any other questions?
Hello, is it possible to correlate ChIP-seq data with RNA-seq data using this pipeline?
Okay, great question. I think as Julia showed in the beginning, we had a method to combine multiple sources of omics data and identify relationships between those. I think we will get to that point again. Thank you. Any other questions?
How to select RNA-Seq tabs? So what's the basis of those selections? Like I have to select this tab, and that tab. Whether I use BWAT or BWAT2T. So what's the basis of those kind of selections?
Okay, so what is the logic behind selecting specific steps?
Okay. So there are different methods that perform the similar task, but generally, I hope that we discussed the logic of the preparation of the experimental data, and so here, each step of the analysis simply follows the experimental step. Just in 2 or 3 minutes, I'll tell you that in reality, several steps can be omitted. For example, preprocessing can be omitted, but generally, this pipeline just goes back from reads we have out of the sequencing machine to initial data. We added adapters, hence remove them. We potentially added PCR duplicates, hence we remove them, and in fact go back to the initial set of short sequences. These reads originated from original transcripts. Then we try to map all these short sequences on genes in order to understand which genes are associated with many segments. These genes are the genes with high expression level. Which genes have no or almost no short segments associated with these genes? So these genes are genes with low expression. So unfortunately, if we now go much deeper into the details, we'll fail to complete even a short overview, because indeed these algorithms have been studied, designed for, I think, more than at least 15 years, and there are great algorithmic ideas behind these algorithms. But currently, we're focused just on overviewing these steps.
Okay, Vladimir, sorry. We have a number of interesting questions here. And just maybe you can quickly help address those. So
Okay.
the question number one was how to know quality of raw FASTQ files before the mapping. Question number two was about the logic of different steps of analysis. Question number three was about making correlations between multiple omics data. Question number four was about identification of isoforms. So I know you probably don't want to address everything at the same time, but maybe you can just give an overview of when those questions would be addressed in your presentation.
Okay, so starting from the beginning, we can still use preprocessing steps and look at the output statistics of what we're doing with. These models provide us a statistics, for example, with the ratio of reads identified as PCR duplicates and hence the reads which are removed. We see the number of reads for which adapters have been trimmed. But anyway, there are ways currently not implemented as modules, as we use them rarely, to look just at mean base quality reported by the sequencing machine. So FASTQ files along with a nucleotide for each position contains an estimate of a quality for this position, and just to look at the number of positions with high quality and low quality, to look at the number of reads containing only high quality bases. It's possible, but another possibility is to get statistics after the mapping, for example, to get number of reads or percentage of reads successfully mapped on genes, to get ratio, to get proportion of reads that had a unique location with respect to genes, and to make a conclusion about the quality of this FASTQ file based on these overall statistics. So primarily number of PCR duplicates that were removed, number of reads that were not mapped, that were mapped uniquely, and that were mapped simultaneously on different locations. So this is a short, well, not complete answer to the first question. So going further about, well, for example, identification of novel isoforms. When we try to identify novel isoforms, we are looking at reads, mapping them not on transcriptome, but on genome, or better to say, on exonic segments of the genome. And these reads can link one exon to another exon, so read can start inside one exon and have the other end of this read in the other exon. So this read just generates a link between these exons. And we have a map of all possible links, so of all exon links supported by reads. Having this map, we can try to identify genes, so combinations of exons corresponding to a fixed gene, or we can find reads, well, logically. Let me avoid explaining here the details. Well, fairly well mapped on a part of a genome that is not currently identified as known coding region. So it's the logic behind identification of novel isoform. So I think these are just short answers to 2 of the 4 questions. Well, let me avoid discussing links between multiple omics types today, as it's really a long topic. So Julia just mentioned that there are some in-house tools we have for these analysis. For example, the association. There are some publicly available tools, for example, for multi-omics clustering. These tools are also collected, well, certain tools are also collected in the platform. So there are multi-omics analysis sections. Well, these are just some possibilities. So multi-omics clustering, some associations, and a number of additional models. But it's just a topic for another discussion. Unfortunately, I'm afraid it will take long even to make an introduction here. And there was the 4th question, so could you please remind it? Okay, if there are more questions.
Yeah, just a second. Any question?
I can also maybe say that we will have a number of breaks further on, and it would be probably helpful if we can move on with the material so that we can do some practical aspects of building the pipelines and understanding their logic. And as we get to the next stage, we can take some more questions there.
Okay, fine. So currently, first, I want to say that there are 2 main types of PCR duplicates. Artificial PCR duplicates, which originate from this step of library preparation, PCR amplification. But there may also be natural PCR duplicates. So while performing this preprocessing step, removing of PCR duplicates, you definitely should keep in mind the details of your experiment, primarily, well, as one of the major parameters, the amount of RNA material you started with, and hence the number of PCR amplification cycles you performed. And especially if you're dealing potentially with short sequences, for example, not genes, but you are focused on microRNAs, then these PCR duplicates removal step should be used with caution or even completely avoided. There are some sources, so here you may find links that state that these preprocessing steps, PCR duplicate removal, adapter removal, well, generally improve the quality of the final expression level quantification, but this improvement is moderate. So preprocessing can be even avoided with getting good results. So let us currently try to discuss what measures for expression level quantification can be used. And the first measure we already discussed is very simple. It is the number of reads mapped on this gene. First, we can work with just raw counts, the total number of reads mapped on a gene. A slight problem here is that some reads, in many cases, an essential amount of reads are mapped simultaneously on different locations. So we may want to split this one count associated with one read between all the locations to which this read has been mapped. The simplest way is to split this one count uniformly, but in many cases more complex statistical models can be used, and for example this RSM module we added to the pipeline uses a relatively complex Bayesian approach. So the idea is that we can perform this splitting of counts associated with the same read in a more sensible way. But in any way, if we're dealing with counts either raw or so processed, so-called expected counts after this splitting, what we may have two genes, and for one gene we'll have many reads mapped on this gene, and for another gene we may have much lower number of reads mapped on this gene. Can we say that, well, natural expression level of the first gene is, for example, expression level measured in the number of mRNA molecules associated with the gene? Can we say that for the first gene, this expression level is higher as we have higher number of reads? Unfortunately, no, and the reason is very simple. If gene is longer, so then shattering this gene or mRNA molecule associated into parts will simply give us more pieces. So longer mRNAs give more pieces in comparison with short messenger RNAs. But the number of molecules we have is the same. So in order to get, well, meaningful quantification, we should keep in mind the length of a gene, and we may perform norming with respect to this length of gene. Another idea behind scaling, behind norming, is the following. Imagine that we have one sample, we sequence this sample, and we then repeat sequencing just the same sample with adding one additional cycle, with adding just depth of sequencing. I mean, generating twice as much reads. So generally, we'll have twofold increase in the number of reads mapped to each genes simply as we have more reads into consideration, more genes in our collection. But it's just the same sample, and it's natural to have the same estimations of expression levels. That is why we should also keep in mind sequencing depth, for example, measured in the number of reads mapped on transcripts. And that's why we use another scaling item, total number of map reads. And it's just a very standard unit for expression level quantification, fragments per kilobase per million map reads. So the number of reads is scaled by gene or transcript length and by the total number of reads we have. Another usual unit for expression level quantification is TPM, transcripts per million. TPM units are for a fixed sample, only for a fixed sample, are simply proportional to FPKMs, but the coefficient of the coefficient C here, the coefficient of proportionality, is selected in such a way that the sum of all TPMs for a given sample is exactly 1 million. So let us try to briefly discuss the logic which stands behind this unit TPM, as unlike FPKM, TPM have a very natural biological interpretation. So let us consider the following amount, number of reads mapped on one gene, multiplied by the mean read length and divided by gene length. So let us try to discuss what is the sense behind these formula. Number of reads mapped on one gene multiplied by mean read length. So what we have in the numerator here, number of reads multiplied by mean read length, is in fact the number of sequenced nucleotides associated with this gene. So number of nucleotides associated with this gene we have after sequencing. After the division, after dividing this product by gene length, we in fact get number of molecules, number of messenger RNAs associated with this gene or with this isoform that we sequenced. If we sum these quantities over all genes we have, in fact, we simply get the total number of molecules of messenger RNA after sequencing. And if we take this amount for one gene and then divide it by the total amount, we have a fraction of messenger RNAs of transcripts associated with the gene of interest in the whole totality of sequenced molecules. So it's just a fraction. Multiplication by 1 million gives us scaling, so TPM tells us the following. In each million of messenger RNAs in the sample, TPM is the amount of molecules associated with the genes we are looking at. So for example, if TPM is 10, then out of each million molecules, approximately 10 are molecules associated with this gene. And another question related to quantification of expression levels is scale. All these quantities, read counts, FPKM, TPM, are the quantities measured in so-called linear scale, so original scale. For the majority, for many types of the analysis, these amounts, these linear scale amounts, are log scaled. So we take logarithm. There are multiple reasons for this transformation. For example, one of the main reasons is related to the statistics, as for the majority of genes, the distribution of expression level is log normal. So after taking logarithm, we get normal distribution, which is very good for the analysis in terms of classical probability theory and mathematical statistics. Another reason is that linear scale is simply similar for dealing with. For biology, relative differences in expression level are more meaningful than absolute differences. If we say that the difference between expression levels is 100 TPMs, it will say almost nothing. But if we say that the expression level increased twofold, threefold, tenfold, it gives more information. And log scaling simply moves these relative differences to differences that can be obtained by taking standard arithmetic means and differences. So, for example, in log scale, twofold difference is simply a difference by 1 log 2 unit. Difference equal to 3 log-fold units is equivalent to the difference, to the 8-fold difference in the initial scale. The problem with going to the logarithm scale, very well, not essential problem, frankly speaking, is that logarithm is defined only for numbers greater than 0, and for many genes the expression level is just 0. So we can't take logarithm, we get minus infinity. That's why a small shift is introduced, the values of shift vary, and one of the standard values is simply one of possible values, but it's not the only option. In any case, the differences in this shift affect only genes with low expression level. And we should keep in mind that for low expressed genes, quantification just experimentally is inexact. So we can't be sure that expression level of low expressed gene is measured really precisely. That is why genes with low expression level in many cases are simply removed from the analysis. But log scaling is also a standard step of the analysis. So running the pipeline we discussed simply gives us estimates of expression levels in the units we set for the samples we uploaded to the platform. In order to see results and in order to compare the results for different types of preprocessing, including or not including PCR duplicates removal, including or not including trimming adapters, you may look, now won't do it, at the files you have in the subfolder output files. There's the next level subfolder, RNA sequencing comparison, with an Excel file. So for two samples, here you have just FPKMs, linear scale FPKMs. For these two samples, just without any preprocessing, with preprocessing that includes only adapters trimming and low quality end streaming, and with the preprocessing we discussed. So shortly, if we compare results for the cases no preprocessing versus no duplicates removal, or even for the other case, you see that for the majority of genes, gene expression estimates are roughly the same. So, for example, for these genes, along the x-axis, we have expression level quantification obtained by one type of preprocessing, and the y-axis gives expression level estimate for the other type of preprocessing. So for the majority of genes, we get the same or approximately the same results. But there are some transcripts for which the difference is really high. It seems, for example, that the dot I'm pointing at is not very far from the line x equals y, but the difference is more than 1. And this difference of more than 1 is a twofold difference, as we're dealing with log2 scale. So mass data experiment gives us the general view, but for the individual genes, for the individual players, we need additional verification, we need additional validation if we want to deal further with some small subset of genes of isoforms identified at this mass experiment. So in any case, this analysis simply gives us huge tables. So it gives us dozens of thousands of numbers for each sample. If we perform mapping on only on known transcripts, we have these numbers only for known ones. If we add modules for the identification of potential novel transcripts that definitely need additional wet level validation, then we have estimates, as you hear here, transcripts marked with X log beginning, these numbers additionally for potential novel transcripts. But the question is what we can do further with this amount of data. These expression level tables are not the end of the story, they are just the beginning of the story. And so now we'll just start discussing what we can do with these data. So let us make a decision. Do we need a break here or?
Yeah, please let us know if we need a break now in the room there. Do we need like a bathroom break or a drink break, a questions break? Okay, I will put it as a no break.
Oh, okay. Okay, so currently we'll just try to see what we can do with the table, with these tables. We'll use real tables for the cell line experimental data Julia briefly described. Out of these dozens of thousands of genes, we removed okay, so break, short break.
Okay, Sahil.
Hello.
Yeah. Okay.
Yeah.
Well, I just noticed that the microphone actually doesn't work that great when you take it far away.
Is it fine now?
It's fine, but I think the farther you go from the computer, the less it works.
Oh, is it fine now?
Say that again?
Is it fine now?
Yeah? No, it is fine, yeah.
Okay.
Yeah, you can, for now, if you have an urgency and you need to analyze data, you can multiply them, sort of read the data to you, or you can touch the data, either of us. If we are still out of the urgency, then if we can put it in devices, but if everyone has a urgency,
I can play
It will take long. Yeah, so
Sorry, yeah.
Okay, so let us know when we should start again. There was a request from the audience to see the speaker on video, especially when there are questions. So we are answering to those requests. Okay, should we continue then? Okay, so is Sahil back there?
Yeah.
Okay, so we will start again. Please continue.
Most of them are back.
Okay. Okay, so currently we'll try to see what we can do with these expression tables, and we start with a very simple analysis. So we have several, well, about 50 cell lines. All cell lines originated from patients with breast cancer. And it's probable that all these lines are similar to each other, as we're dealing with cell lines associated with just the same pathology. But how can we know if it's true or not? It's really impossible to go through all these numbers just by eyes, and our visualization stops at the dimension equal to 2 or 3. So if we have 2 attributes, we can visualize our data as dots in a plane. 3 attributes can provide us association of samples with dots in a 3D space. But we have thousands of genes, tens of thousands of genes. And so this visualization looks impossible. However, we can try to find an optimal plane that keeps as much data as possible and still provides us an opportunity of visual inspection of the data. So currently, I'll try to explain what I mean using a very simple example, and then we'll return to the presentation. So we have a simple pen. So in fact, what you see in your screen is a 2D projection of a 3D object. If we choose a plane of projection in a poor way, we have very poor understanding of an object we're dealing with. So we have just the same object, but we cannot understand what we're dealing with in this projection. Changing the plane, changing the projection gives us better and better way of seeing these objects. For example, in this projection, you can really understand what is the object. Okay. Okay, so if there are any problem with video, please let us know.
I think it is working fine, at least for me.
Okay.
It's working.
Okay, great. So, and a very simple thing can be applied to the data we're dealing with. We can try to find optimal plane or optimal 3D space or optimal planes that can help us in visualization of the data we have. So let us try to test the thing I'm talking about. So let us create and run our pipeline and inspect the result. So we'll be working with unsupervised analysis, as in this analysis we know nothing about data underlying our samples. We think we start at least with the understanding that all samples are similar, they originated from the same pathology. So in order to perform this simple kind of the analysis, we select unsupervised analysis section, upload expression table for the analysis. This is the file cell lines expression data.txt. We'll take a look inside this file in a minute or two. So we select cell lines expression data file. Okay, so main files in the folder we're working with today, and the file cell lines expression data. Okay. Okay, great. So we select this file, cell lines expression data, txt, and press open button. So this file is uploaded to the system. Unlike the previous analysis, here we indeed take the file from your computers to the system. So it may take several seconds. Please let us know in case of any problem. So again, we add file cell lines expression data. So that's it. And go on. Continue. Continue. Start. Then our principal component analysis module is PCA draw, as we want to have plots as an output. PCA draw. The default options here are okay. In order to get additional help regarding these options, if you need explanation, you can just point on I zones and have short comments. But here we say that we want visualization associated with 3 first principal components. We need no scaling, centering, and transpose explains which information goes via rows and which via columns, samples and genes or isoforms expression. So here we are. Then it's just the end of our pipeline and pipeline name. See draw, for example. Save the name. Save the name. And then after the name is saved, simply run this pipeline by pressing Run Pipeline button. That's it. So the pipeline is starting. It may take several seconds. When the pipeline is started, you can monitor its progress here or in My Pipelines. My Pipelines reports the results and everything for all pipelines you previously had. So again, pressing on the name brings us to the screen with the pipeline progress. While the pipeline runs, let us take a look at the file we uploaded in order to understand what we have inside our system. So what is this file? It is a very simple file in which for each sample, for each cell line, and for each genes out of, well, about 7,000 genes, we have expression. Here, expression is measured in log-scaled, log2 scaled FPKMs. So we use FPKM units with log2 scaling. And so initially, each cell line is a point in a space with a number of dimensions close to 7,000. Well, I reload it to see the progress. So for me, almost completed. And in order to understand the simple picture, we project all these data. We project all this data simply to a 2D plane. If you reload the page, you'll see the progress. It won't take too long, but it may require several dozens of seconds or a couple of minutes to complete in this platform mode.
You will also get an email notification after the pipeline is finished.
Okay, for me it's done. So let us additionally wait for some time. So reloading updates the progress indicator, and after the pipeline is completed, you see 100% done signature in the top right corner. So if you have the pipeline completed, you may take a look at the output. The main output of the pipeline is stored in the download pipeline output files folder. So in the left folder, you see here download pipeline output files. These are the main output files. So go in here. For example, we have a PDF file, PCA plots. So this is the visualization we wanted to have. So I download this file and open it. Each dot is associated with one sample. And we started our discussion from the fact that all cell lines originated from the patients with the same pathology. And we wanted to see whether all cell lines are similar. However, here we see at least two groups of samples. We definitely see a clear separation of samples into two groups. Probably somebody will see additional separation into groups in the left part or in the right part. At the same time, we know that there are multiple subtypes of breast cancer, and we may hypothesize that this separation is related to the subdivision into subtypes. And the authors of this study were working with the author of the study that produced the data, classified all the samples into four classes, into four types of the breast cancer: luminal subtype, basal subtype, Claudin low subtype, and normal-like subtype. So in fact, we have information about clinical subtype for each sample. And probably it would be interesting to see how subtypes are located in these plots. Well, we can describe the picture, but a better way to look at it is to generate the picture. In the same folder, in the main files folder, we have an additional file, expression data marked. It is just the same file, but for which each sample is marked by a subgroup. So let us repeat generating PCA plot, but upload this file, cell lines expression data marked, in order to get different colors for dots associated with different subtypes. So the analysis is just the same, but the picture will include information about the predefined class. So in order to do it, we create one more pipeline, just the same one. We used unsupervised analysis. In this unsupervised analysis, we upload the file, but this time we upload the file expression data marked. We upload the file expression data marked. That's it. So again, the difference is that just here we have predefined classes for samples, and we'll see how these classes are located in PCA planes. And all the same steps are the following. It's a simple analysis. So we upload the data. We upload the data, and then just repeat the steps we did. Continue. Start. PCA draw, for example, with default parameters, and give a name to the pipeline. And run it.
Okay, maybe this is a good time to see if everybody is following along and if there's any questions that we have.
I have this question that if in the first column, instead of the transcript ID, you have the gene names, would this pipeline work on it? Or the data needs to be in this particular format of having samples versus the transcript IDs?
Did you get that, Vladimir?
Oh, could you please repeat the question, because the audio is a bit worse than I wanted it to be.
Yeah, as I understand, the question is about formatting the input file for the PCA. How do you have to organize your transcripts and samples?
Okay,
so rows and columns.
Thank you very much. Perfect question. So here we have multiple options. We can first, for the PCA, for the principal component analysis, transcript or gene IDs are simply ignored. So in this type of the analysis, we have features, but feature names are not essential. Then we can arrange samples by columns and features by rows, or vice versa. And all we need, let me show you. I'll just show you it on the screen. I upload, well, any file. All we need here is to tell system which goes by rows and what information goes by columns. In our case, when samples are columns, we set transpose equal to yes, and in the opposite case, we simply set transpose equal to no. And if we have additional information about classes or groups or something like that, we simply add a line marked by a keyword group here. So here the keyword is grouped with the name of a group. So again, here two options are possible. What goes by rows, what goes by columns. Both options are possible. So, okay.
Are there any other questions?
So meanwhile, I've got a question to you. So if we look at the previous PCA plot, here it is on my screen, and remember that there are 4 subtypes. So what's your idea? How these subtypes would be located? 2 on the left and 2 on the right, or something different. So can we make a prognosis looking at this picture? So just your feeling.
Sahil, can you be the voice of the feeling?
Yes.
And so what does the voice of the feeling say? So frankly speaking, I also have no hypothesis looking at this figure. But anyway, let's look at the correct answer. So hopefully the pipeline is completed for you as well. So please let me know if I'm wrong. So for the next pipeline, we'll simply have a look. We'll simply have a look at new plots we have here. And what we see is that the luminal subtype is very well separated from 3 other subtypes, again PCA plot, but these other subtypes are relatively close to each other in the first plane, principal component 1 versus 2. We set the number of principal components to be drawn to 3. That is why we also have planes associated with principal components 1 and 3, 2 and 3. And here in the next plane, you see that cloud and low subtype is well also separated from basal and normal-like, but basal and normal-like subtypes look similar. Anyway, please pay your attention to the fact that initially we knew nothing about the subtypes, but this analysis provided us an unsupervised but very clear, very definite separation into at least 2 groups. But this is a visual inspection, and visual inspection can hardly tell us what is a normal, well, an acceptable, a good number of clusters of groups for a given set of samples. So here we see that at least 2, but if we knew nothing about true subclasses, we could not set the number of subclasses to 3, 4, 5, so on, based just on our visual inspection. So let us create one more pipeline for the unsupervised analysis of the data. It's just in the same section for performing formal clustering, in our case hierarchical clustering of the samples we have. So again, we go to the unsupervised analysis section. Again, we upload the file expression data. This time we do not need marks, we don't need information about subtypes, about classes. That's why we again upload the file cell lines expression data. So again, it may take some time. We start creating pipeline, and this time we want to perform clustering, for example, using hclust, using hierarchical clustering model. So hclust. Here it is. Here it is. Lisa, here, let me know if everybody's here. As here, we'll set a bit different options. For example, we'll set the expected number of clusters to 4. If you like 5 more, please keep 5, absolutely not a problem. And let us change linkage type, for example, towards D2. So it's more important because linkage type is an essential parameter of hierarchical clustering. So linkage type is changed to, for example, towards D2. Definitely, it's not the only possible option. And that's all. That's all. Again, do transpose simply tells which information goes by columns and by rows. That's it. That's it. So we give any name to the pipeline. And start it. Great. So while it's running, let's briefly discuss what is performed by this pipeline. So initially, we know nothing about the number of groups. So initially, we look at our samples, which are here illustrated by dots in a plane, and associate one group with each individual sample. So initially, in the bottom-up approach of hierarchical clustering, we have the number of clusters, of potential groups equal to the number of samples. Then we simply unite 2 samples, which are the closest, into one group, and treat these samples as an inseparable, well, meta sample or quasi sample. So for us, Currently, it's one meta sample, but not two individual samples. So it's one dot instead of two dots. And then we continue the same process. We look at two samples or meta samples which are the most close to each other and unite them into one group. So at each step, the number of groups is reduced by one. We continue the process and we unite the closest pair of samples into one groups. But here, well, initially, we need to set distance between individual samples. And here, we need to define what is the distance between meta samples, between what is the distance for this situation when at least one argument is not a standard sample, but a group of samples, well, quasi-sample. So this distance can be defined into very different ways. Minimal distance between samples from the first group and from the second group, maximum distance, mean distance, or more complex definition. So it is the question, so it's the fact determined by the linkage type, how to define the distance between these quasi-groups, what formula is used. So this process goes on and on and on. At each step, we reduce the number of groups by one and finish this process by one group that unites all the samples. At this visualization, it's hard to understand the logic, it's hard to understand how the process goes on. But there's one more standard visualization for the result of the hierarchical clustering, so-called dendrogram. So let us have a look at the progress of our pipeline. Well, let's inspect what's going on there. So the mine is done. So please let me know if yours is done as well. So if it is, again, if we want to inspect our results visually, we have a PDF file, cell lines expression data series hclass.pdf. And here you see the results. Samples, in our case, breast cancer cell lines, go into the line below. And step by step, these cell lines are united into groups. The height at which two subgroups are united shows the measure of dissimilarity between these two groups. For example, if grouping is performed at a low height, then the united groups are similar. If we unite two groups at high height, it means that groups are not similar at all. So this figure clearly states that we have two well-defined clusters, and potentially both clusters can be further divided into two subgroups. But if you have a close look at this figure, you'll see that these groups won't just coincide with the subtypes we have. For example, one of our subtypes, normal-like subtype, is located in the rectangle you see marked as green. So if we cut this tree into two groups, we need to select the cut height at the level of 300, 400, we have two groups. If we cut the tree at a low height, we get four groups. If we continue reducing the cutoff height, we get more groups, but please take a look. What will be the next separation if we reduce the height to the level of approximately 200? Here you'll see that HCC 202 cell line will be identified as a separate cluster, as a separate outlier. So just the same cell line that was identified by the analysis explained by Julia. Anyway, four clusters are seen really clearly in this figure, and there's no wonder that luminal subtype here is subdivided into two classes, as indeed it includes two subtypes of the second level, luminal A subtype and luminal B subtype. We obtain this division into clusters in an unsupervised manner, looking at all genes. After very simple prefiltration exclusion of genes with constantly low expression level. The question is, can we get more looking at lower number of genes? So can we get more information looking at much lower number of genes? It seems that we lose many information, limiting our focus on just a few genes. But at the same time, if we analyze large sets of genes, the majority of genes, the majority of transcripts simply introduce the noise. So there are only some genes related to the question of interest. And reducing the number of genes we're looking at can potentially improve signal-to-noise ratio. Indeed, we lose some information, but still we can keep much information and greatly reduce the level of noise. For example, let us look what can be done just in a similar analysis type if we limit our attention on 15 genes out of our initial set. So let us create two pipelines similar to the ones we just created, just the same types of the analysis, but limited to 15 genes. Again, we're in the unsupervised analysis section. So first, for the PCA analysis, we upload the file cell lines 15 genes marked in order to see the complete figure. Cell lines 15 genes marked. Here it is. It's small as there are only 15 genes, not 7,000. So PCA draw analysis. PCA draw analysis. Oh, it's the first pipeline we're going to create. And the second pipeline we're going to create in a very similar way is pipeline for each cluster. In this case, we upload non-marked file cell lines 15 genes. Cell line 15 genes, and again perform just the same steps. We upload this small file. And create a pipeline. Each cluster, for example, with 4 clusters. Again, you can keep any other number of clusters, as it does not affect the dendrogram we get. And again, linkage type, the algorithm for getting distance between quasi samples, set towards D2. We save it and go on and complete this pipeline, for example, H cluster and run it. Okay, we'll soon have the results for these pipelines, but in any case, if we look at these tables, we'll again see that for these tables, just a moment, let me show you. For these tables, the selected genes are identified by their ensemble IDs. So information that's potentially hardly interpretable, as only few of us remember the association of specific genes with their ensemble IDs. So if we want to get more meaningful information about genes, gene names, let us run the third pipeline and then just generate the results, just gather the results. The pipeline we're going to run, the next pipeline, is associated with the utilities section. We simply want to know what are the genes we're working with. So we upload our file cell lines 15 genes. Cell lines 15 genes. And use annotation model that will look at the database and add standard gene symbol and go categories to each gene we have in our table. So that's it. Annotation, for example, with the default categories. Okay, so let me just repeat all these steps. The question is how to restore gene symbols and go categories having, for example, ensemble gene IDs. Well, technically, it can be done simply by using the platform, by creating a pipeline at the utilities section. So we upload a file with genes for which we want to know these gene symbols, these go annotations, just the file we're working with, and create a pipeline with the annotation model. It says that for ensemble gene on transcript IDs located at column 1 in a table that has a header, we want to add information including go categories. So that is a very simple pipeline. That's it. Okay. Okay, and let us look at the results. Let us look at the results. What we get, well, please let me know which pipelines are completed for you, which are not. Well, in any case, what we have for these 15 genes. For these 15 genes, we have a much clearer separation of groups on the PCA plane. So is the figure you get out of the PCA analysis. And indeed, we see that going into lower number of genes, well, into the subset of genes we can call a gene signature, we can have clear figure of what's going on. And just a moment. And if we look at the results of hierarchical clustering you obtained, okay, perfect. So if you download the results of the hierarchical clustering, you'll see the dendrogram I'm having here with additional marks. So I just point your attention to the fact that these 15 genes indeed give us clustering into 4 clusters. That perfectly coincides with the division into 4 subtypes. So reducing number of genes with an increase of signal to noise ratio or an analog of the signal to noise ratio gives us the results perfectly concordant with what we're dealing with. So that's hierarchical clustering. And finally, we're waiting for the results of the annotation, which will also give us gene symbols and all the other data type. But it is the result of the unsupervised analysis. It is the result of the unsupervised analysis. Definitely, there are multiple other methods for unsupervised analysis. For example, for clustering, well-known method is k-means. There are other methods for clustering that may produce results different from the hierarchical clustering and that may require different input. For example, for k-means, we do not have a clear visualization with a dendrogram and have to predefine k number of clusters, number of groups in advance. But this initial analysis linked with clinical data, linked with phenotypic data, gives us separation of our samples into groups. And we have another task, having the separation of samples into groups, how to identify the group for new samples. For example, having our data set as a learning set, we identified that breast cancer patients can be divided into subgroups, and how to define, how to understand what is the subgroup for a new patient. It's the question for the supervised analysis. So probably at this point we may have a short break, well, if you're a bit tired. And finally, at our final part, before a short hands-on practice, we'll briefly discuss the analysis in a supervised problem statement when we initially have this division into subgroups that we could identify using unsupervised analysis.
Okay, so as I understand, in about 15 minutes, some people will have to be leaving.
Okay, so in this case, we can simply try to have a simple example which will show you that indeed these methods of the analysis can simply be used to analyze a data set you know nothing about initially. So what I'm talking about is the following. In the set of files you have, there's a subfolder hands-on. What is this set of data we have in this file? we took 2 additional projects, both dealing with breast cancer, but in one project, the samples were human samples, just biopsy samples, direct biopsy samples or tissues taken in frames of surgeries. And the other data set is xenograph, is data originated from a patient-derived xenograft project. So human breast cancer was implanted into mice and then taken again out of the non-human mice microenvironment. And it's clear that microenvironment in human and in mice should be different, and the samples should also be different. So we took just the same 15 genes to make the analysis faster, but the samples here are intermixed. So some samples are associated with humans, some associated with mice. So could you divide this set into a human set and mice set, probably not naming each one, but telling which samples belong to the first and which samples belong to the second, using the automatic modules we've discussed. So in order to understand that this analysis is really simple, in spite of the fact that the number of the amount of information is relatively large, well, dozens of samples and several genes. So the task is to separate this data set into 2 different, really different subsets: mice and human-derived samples. Is the task clear?
So as Sahil is walking there behind, Sahil, just let us know if everybody is following, if everybody has seen the file. It's in the original zip folder that you downloaded. There is a file.
So hands-on, and the only file in the folder hands-on, file with intermixed data sets, human-derived and mice-derived.
Okay, and people that are just joining online, you can also let us know on chat if you are following along or if you're having any issues. In the meantime, I just want to mention also, Vladimir, there is somebody requesting clarification on the steps for the annotation pipeline. So if at some point we can review that again, that would be helpful.
Sure. So probably a good option is to give several minutes to implement this hands-on task and then return to the annotation pipeline. Okay, if you have started your pipelines and just waiting for the results, well, please let me know, and we can briefly discuss this annotation and then to go through some additional topics very briefly. Okay, so again, regarding annotation, here currently we're talking about very simple annotation about taking ensemble gene or transcript IDs and adding information which is simpler for manual interpretation. I mean gene symbols, so just standard gene names, and gene ontology terms, cell localization, gene ontology functions, and so on. And in order to perform this pipeline, definitely all we need is the set of ensemble gene or transcript identifiers. In our case, these identifiers are located in the first column of the table. So we simply upload this file. This one, for example. And tell that we need to add information for identifiers stored in the first column of the table, that the table has a header, so the first row is simply a header, and we want to include gene ontology data into the output. That is all. And the output for this pipeline, I hope you also have an output for this pipeline, includes a table marked with out suffix, so it is just an initial table into which in the right gene symbols are added. So yeah, for these ensemble genes, gene symbols are added. And here you see that in our subset of 15 genes, there is, for example, a very well-known epithelial cell adhesion molecule, EPCAM, is present. And then Go terms are associated with each gene. For example, for the case of this well-known cell adhesion molecule, the cell terms include plasma membrane as its adhesion, lateral plasma membrane, integral component of membrane, and others. So for example, DC and protein, these Go terms include function, wound healing as a biological process. And along with gene symbols, here we see a bit fuller description, full gene name, for example, collagen type 6 trimer, etc. So it's a simple, very simple annotation limited to adding gene symbols, gene ontology annotation. So if you're ready with the hands-on task, we can discuss the results and then briefly discuss just a couple of more topics.
Vladimir, there's also a question here. If you will, after this hands-on work, go over a standard differential expression pipeline with full annotation.
Exactly. So again, and asking Sahil as the voice from there, what are the approaches you tested in order to separate the data set into two subsets? And what results did you get? Any ideas?
You're also welcome to write your ideas on chat, and we'll announce them here.
Okay, so both options we discussed today, principal component analysis and hierarchical clustering analysis, both of them can solve the problem we have here. For example, if we performed hierarchical clustering for this file, well, for example, I'll show you the result with 2 clusters in an output. Well, we'll see what's happening. And principal component analysis potentially can also solve this task, but the output will be a bit longer to interpret. So initially, we have groups completely intermix.
Okay,
Now,
we do have an answer here from the audience. Somebody said PCA with 2 groups.
exactly. So PCA gave 2 groups. So let me just take a look. Okay, for example, as we're waiting for the result, I'll run this pipeline as well, as it's really fast. Great. Great. So just a short waiting. So what we have for hierarchical clustering as an output here? We see 2 well-defined groups, and the answer for this simple task is the following: all samples marked with odd numbers belong to 1 group. All the samples marked with even numbers, 2, 56, 60, etc., belong to 2 groups. So this separation is just because indeed one subset is human-derived and the other subset is mice-derived. And in human-derived samples, all the samples belong to the same subtype. So that's why the unification height is relatively low. And in the mice-derived subset, in PDX subset, there were 2 types of breast cancer, and these 2 types are defined by these clusters that can be obtained by cutting the tree at the height of, for example, 30. So indeed, again, hierarchical clustering gives us a clear separation. And if we look at the results of the principal component analysis, after we get at it, it will take just a few more seconds. We'll also see this clear separation. For the hierarchical clustering, definitely we have not only the plots, but we have additional information. For example, we have a file with the suffix clusters only.txt, and just a moment. What is the content of this file? I'll open it in Excel to have a simpler view. So for each sample, this file gives a cluster identifier. We set the number of clusters equal to 2. So the cutoff height was selected in such a way that the resulting number of clusters is 2. If we've set the cutoff level to 3, 4, etc., we'll have the same number of clusters here. And looking at samples united in one cluster is very simple. We just look at samples marked with the same cluster ID. And in the PCA plane, we indeed also have a similar separation into two groups. One group, the left and bottom, and the other group, top right. So it is the separation. So having the groups, we can think about the next problems, and one of the problems is to find genes that have different expression in these subgroups we're dealing with. So first we need to define, we need to analyze to understand what does it mean a gene has different expressions in two compared classes, in two compared subgroups. First, we may look at mean expression levels. Different expression means that average expression levels are also different. But look at the example pictures to the right of this slide. You definitely see no differential expression in the uppercase and evident differential expression in the lowercase. In the blue group of samples, expression level is evidently much higher than in the red group of patients. At the same time, the difference between means in the upper and in the lowercase is just the same. 1 in log 2 scale, so twofold difference in the initial scale. That is why fold change, the difference between means, can be used as the only parameter, as the only characteristic of differential expression. We also need to consider statistical significance of this differential expression obtained using one of the standard or non-standard statistical tests. In the simplest case, it is a student's test, or so-called t-tests, that compares the difference between average levels of expression levels compared to deviations, compared to variations inside expression levels in each group. More complex cases, which are better for small sets of samples, use more complex statistical schemes. For example, statistical schemes based on negative binomial distributions are popular. Technically, in order to get the set of differentially expressed genes, if we want to use a simple test, for example, t-test, we can do it even taking expression levels provided by our previous pipeline, one of our previous pipelines, as an input. But as I told, more complex statistical approaches are preferable. So in order to use more complex statistical approaches, let me just show you the pipeline design. And we can create this pipeline together. All we need is to upload samples we want to compare. For my case, I'll use another set of links to files that are already present in the platform. Links, cloud and low versus normal-like samples. The above 6 samples in this set are cloud and low breast cancer cell line samples, and lower 5 samples are normal-like samples. But in this case, so in this case, we want to compare expression levels in 2 groups. So we want to find genes that have higher expression in one group and lower expression in the other group. So we need to specify which samples belong to which group. There potentially can be more than 2 groups, but let us limit our consideration to 2 groups. So, for example, as I told, in this case, lower 5 samples belong to one group, and above 6 samples belong to the other group. And if we initially know the difference between which sample belong to which group, here we press continue as we have groups. Well, submit changes in groups and create a pipeline similar to the ones we initially had, as here we again start with raw sequencing data with FastQ data. So I just recreate the pipeline we had initially. But here at the end, we want to find the set of differentially expressed genes. It can be performed if we want to use more complex statistical models based on negative binomial distribution, for example, by well-known HR or DSEC2 modules. So technically, that is all. So we give a name to the pipeline and run it. Regardless of the module we're using, what we get as an output. So annotated output for these pipelines is provided in the subfolder output files. Then we can go further into the differential expression subfolder. So the outputs have similar meanings, and I'll pay your attention to the main information. Just a moment. Sorry, just a moment. So let us take any of these. What information can be used here? So let me sorry, just a moment. Okay. So what information we have after the differential expression analysis? For each gene or for each transcript, we have fold change, potential, mainly log fold change. The sign shows in which group the expression level is higher. Then we have statistical significance of the differential expression, the probability that we'll have the same or even higher fold change in the case when in reality there's no difference in mean expression levels. Adjusted p-value, which is the main parameter that is examined if we want to look at the set of really differential expressed genes, and the base level, so the mean expression level in both groups or the mean expression level in just one of the groups. It depends on the instrument we're using. So it's the standard output of any tool for the analysis of differential expression. So we'll get just the similar output for DSEQ2, DSEQ, HR, etc. And what are, first, do you know what is the reason for adjusting statistical significance, for adjusting p-value in this case? So why do we need to additionally add some adjusted p-value, which is also denoted as family false discovery rate, FDR, Q-value, etc.? So the reason is the following. It can be explained by a very simple example. Imagine that you have a coin, you drop this coin 10 times, and 10 times it falls with the same side up. So it is suspicious. The probability that it's a standard coin and it dropped 10 times, all 10 times the same side up, the probability is low. But if we repeat this experiment for several thousand times, we'll definitely have several coins with this behavior. It does not mean that these coins are not normal. It potentially simply means that the number of tests we were simultaneously running, the number of coins, was too high, and what we see is associated not with individual coins, but with the number of tests. So this adjustment takes into account the number of tests, the number of genes we're dealing with. And in order to select differentially expressed genes in a standard sense, so what are the standard thresholds? Standardly, a gene is called differentially expressed if adjusted p-value, Q-value, FDR, does not exceed 0.05, that is 5%, and standardly has fold change of at least 2 times, so log fold change of at least 1, so higher than 1 or lower than -1. These are the thresholds that are not universal. In different types of the analysis, more strict thresholds can be used, for example, 0.011% for adjusted p-value, and more severe threshold for the fold change. Additionally, in many cases, lower expressed genes are simply eliminated from the analysis, as we initially discussed that the error in quantification of expression levels for these genes is much higher. But anyway, the result of this analysis is the table, which further allows you to select main players in the field of differentially expressed genes. So the main parameters are fold change, statistical significance, mainly measured in adjusted p-value or false discovery rate as another name for the same. And additionally, it's a good idea to exclude genes with low expression. After having this analysis, we may think about further biological interpretation. We may try to think about the following question. Is there any pathway? Is there any pathway? Is there any, well, experimental group of genes overexpressed by differentially expressed genes? Can we say that in our experiment, genes associated with a particular biological sense increase or decrease their expression? And there are multiple approaches to try to give an answer to this question of interest. So the first idea is very simple, and it is utilized, for example, by the well-known David online service, David service for gene enrichment analysis. Let us simply take a set of genes with the highest level of differential expression, with the lowest adjusted p-values and highest fold changes, and simply forget about these additional parameters. Well, let us simply take these genes and let us try to analyze whether these top genes, well, say 100 or 1000 genes, are enriched by genes belonging to a particular pathway. What does it mean? This gene set is enriched by genes belonging to a particular pathway. It means that if we take random genes with a number equal to the number of genes in our pathway, the probability of getting the same number of genes belonging both to the pathway and to our set of differentially expressed genes is low. So, for example, in our set, we have 50 genes belonging to this pathway. And in reality, if we take the same number of genes, the probability of having 50 or more genes is low. So that is the approach used, for example, by David as the first generation gene enrichment tool. The problem here is the following. We ignore too much information. We ignore too much information associated with genes in the results of differential expression analysis. For example, we completely forget about the level of differential expression, for example, about fold changes. Here, losing such a great amount of information leads to poor results of the analysis. The second level of these instruments takes into account this additional information about differential expressed genes, for example, log fold changes. And in the platform, for example, there's a module based on our R-script gauge adopted for a simple use with human experiments. And this tool gives us an opportunity to give a natural interpretation of the differential expression analysis results. So in order to save time, I'll just show you what we get in the enrichment analysis for this experiment. First, just a moment. First, for each analyzed pathway, we get a statistical significance of enrichment. So we see which pathways are really enriched by genes in our set of differentially expressed genes. Standard parameter used is adjusted p-value, that's in many cases denoted as q-value. So here we see that, for example, focal adhesion, calcium signaling pathway are enriched by differentially expressed genes. And if we're looking at standard pathways, for example, KEGG pathways, we also have a natural visualization of differential expression. We have pathway in which players are colored, and the color code is based on the fold change level. Genes differentially expressed in one direction, for example, downregulated, are marked in one color, and genes with the opposite regulation are marked with another color. So this analysis is non-trivial from the statistical point of view, but it is trivial from the technical point of view using the platform. So all we need to do is to upload the file, but here please take into mind that for this type of the analysis, if we want to use the complete information about fold changes, we need to upload the whole table, not only differentially expressed genes, but the whole totality of genes in the experiment, as the method requires all information as the background for its statistical model. And the output is just what I'm showing you. So technically, this untrivial analysis can be performed simply in the case of Tauber Bioinformatics platform in the utilities section by using the human gauge model. Unfortunately, it seems that the time is coming to its end, but you've got all the files for testing it, and I definitely will be happy to answer your questions if you'll try to test it and if you see that certain parameters seem to be unclear. So you have a form here to present your questions, and we'll definitely try to answer them. So yeah, as far as I see, probably we're running out of time. Anyway, we'll be happy to try to answer your questions.
But also, you can let us know. I know that some people had to leave. If you don't have to leave, we can just finalize this one last pipeline and look at the outputs if you would like to. There's a few more things to that pipeline that could be shown. But you can let us know. I don't know, Sahil, if you can. Okay, I see that somebody is asking, is it possible to save the file after adapter cleaning, RNA editing, run?
Okay, it's a simple question, and I'll give a fast answer to this question. If we look at the pipeline, any pipeline dealing with these modules, we have final results in the main download section, but all intermediate results are also, well, I'm just looking at the complete pipeline in my stack. Just a moment. Example. Hopefully here it is. Yeah. So all intermediate results are also accessible in the folder jobs temporary files. So for each job, for example, Trimatic, PCR cleaning, there are all intermediate files saved, and they're also downloadable. Okay. So all statistics, all intermediate files are accessible.
Okay, so that answers that question. So the question about what the pipeline looks like with gauge method with the enrichment analysis, we can finish that pipeline and just look a little bit more in detail into the outputs of the pathways if everybody has time. So if you have time, let's just continue. We'll take just a few more minutes. Vladimir, do you want to just demonstrate the pipeline with gauge and
okay, I'll just try to find files we have. Okay, it's absolutely not a problem. So again, let us briefly take first a look at the input for this pipeline. The gauge deals only with two types of gene identifiers, gene symbols, and entrace IDs. So ensemble identifiers should be transferred to something like that. It can be done simply with the annotation section. Probably it will require some time to annotate thousands of genes, but in any case, it's not a problem. So what is the input for the gauge pipeline? It is just the output of the differential expression analysis pipeline. So we have IDs, we have many parameters that characterize the rate of differential expression. The gauge takes one parameter as an input. Standardly, it's log2 fold change. And additionally, as I said, gauge deals with gene symbols or interest IDs. So we added gene symbols using ensemble IDs that were initially present in the output. So it's the input. The minimal input, and in fact, the complete input that is used contains two columns: gene identifiers given by gene symbols or interest IDs, and rates of differential expression given, for example, with log2 fold changes. That is the input. And in order to generate this pipeline, we have to go to the utilities section, upload this file. For the DSEQ2 output, this is a file DSEQ2 full output annotated. Annotated here means that we just added gene symbols. For the HR, a simple file is one below. There we have just a slightly different order of columns. So I upload this file to the system. And let us take a look at options we need to specify for this model. The options are quite simple, and comments are accessible. If we point at the I section, we have short explanations. In our table, let me show it once again, we have the header. So column headers are present. Column with gene IDs, where gene IDs are gene symbol or interest ID, in our case, is the second one. And let us remember that fold changes are located in the fourth column. So gene identifiers are located in the second column. Sorry. Prime characteristics of differential expression, for example, fold changes, are located into the fourth column. There are two types of the analysis, parametric or non-parametric. Both are applicable to fold changes if we use statistical significance, Q-value, for example, as a measure of differential expression. Non-parametric statistical approach is preferable. Then we select the set of genes, the set of pathways we're going to look at. For example, standard CAD-MET-SIG is the set of KEGG pathways related to metastatic and signaling pathways. Finally, we set a direction of differential expression that is taken into account. Combined means that we combine up and down regulated genes. Separate means that we separately analyze down regulated genes and up regulated genes. And down and up means that we limit our analysis only to down or up regulated genes. And the final threshold is simply for drawing. We'll draw the pathways with enrichment significance that does not exceed the threshold of 10%. So false discovery rate below 10%. And that is all. So that is all we need for this pipeline. That is all we need for this pipeline. It will take some time to be completed, as the input here, as I said, completes the complete set of genes. Well, here we left even non-expressed genes, so 60,000, but it won't take too long. So probably if you have some additional questions,
Yes, there
then
are.
we'll
So,
see the output just today.
okay, so additional questions. One is, can you draw a curve of the gene set enrichment analysis as an output from this gauge button?
So here I need to understand what do you mean by the curve associated with gene enrichment.
So I have this question from Andy B. I don't know if you're in the audience, maybe you can ask directly.
Yes, so if you just provide a comment, what curve do you mean, I'll give you an answer. So currently we have all these pathways, KEGG pathways, standardly visualized KEGG pathways with color codes of differential expression and the text table. So currently no additional curves are included, but in the majority of cases it's not a problem. In any way, for multiple modules, including this one, we output not only the results of the processing, but we also output our scripts that are used inside the platform to provide the analysis results. So what we also have is an R script that performs everything from taking initial data to producing the plots. So if any additional information is required, this R script can be used as a base, and it can be slightly edited, for example, by a user, if a user knows basis of coding, to get any additional information available inside this type of the analysis.
Okay, thank you. There's another question here. In what situations do you use supervised, and in what situations do you use unsupervised machine learning methods?
Okay, thank you very much for this question. The difference is in problem statements. So these methods provide answers to completely different questions. In the first case, we know nothing about separation of our samples if we analyze samples into subgroups. So we just have a set of samples, and the task is to understand whether this group of samples can be subdivided into any, well, more or less natural subgroups. We do not know this division, but in this type of problem statement, we try to find this division. So in any case, unsupervised analysis means that we know that we have no additional information about these samples. In the supervised analysis, we initially have information about samples, for example, about classes of samples, about subgroups of sample, and the question is not to divide the group of samples into new subgroups. We already know the subgroups, but the question is the following: how can we tune a classify function that will take new samples as an input and attribute these new samples to one of the groups we initially had? So we had patients, we already divided them somehow into subgroups, and then for new patient, we want to attribute this new patient to one of the groups we initially had. This is the problem for the supervised analysis type. Okay.
One question from the audience.
So how can we analyze the biological replicates together?
So again, problem with order. Could you please repeat your question?
How can I analyze the biological replicates?
How can you analyze the biological replicates?
Okay, sorry. So what about replicates?
How can you analyze the biological replicates?
Well, it depends. Again, it depends on the problem we're dealing with. In our case, it depends on the experiment you're working with. I won't give you the universal answer here, but in a way, in many analyses, biological replicates can just be grouped into one subgroup or group of samples in the analysis. So we can account this information that a set of a number of samples are simply biological replicates by united these samples into subgroup. But again, a lot depends on the design of the experiment, of the number of macro groups we have, number of different conditions we have. We definitely should understand that there are independent samples, there are biological replicates for some conditions, for some effects, and moreover, in multiple experiment designs, there are technical replicates. We definitely should take this into account and do not treat technical replicates similarly to treating independent samples. But unfortunately, I don't know the universal answer here. It depends again on the macro design of the experiment.
Okay, we have another here. Can the platform perform whole exome analysis?
Just a moment. I'll try to give an answer visually. So again, it depends on the goal of the analysis. The general answer is yes for many types of the whole exome analysis. For example, for the identification of SNPs, of copy number variation, there's a section that performs this job. Currently, we're trying to update this section to add some modern approaches, but the formal answer to this question is yes, there is such a section.
Okay, thank you.
Can you analyze the monkey genome?
What was that? I did not hear that.
Using this pipeline, can you analyze the monkey genome sequencing data?
Other genomes, non-human genomes?
Yeah, non-human genome.
So basically, we have several standard model organisms here. So you see the list starting from Arabidopsis and finishing with, relatively rare types of genomes. It's not complete. These are just the genomes we had to work with. But adding a new genome is a relatively simple task. So if there's a project or there's a user who needs to deal with another standard genome, this genome can be added to the system and added to all these forms. Currently, I see that the gauge pipeline is completed. So as we discussed, the output here is gathered in one zip archive, just in order to avoid multiple files. And the type of the archive, the type of files here is the one we discussed. We have a simple set of pathways with enrichment statistical significance associated with each pathway, and visualization for enriched pathways. Okay.
Okay, so just to summarize, I know that people have to leave, but just to summarize, we were able to go through several sections. Next generation sequencing data pre-processing, including trimming technical sequences and removing PCR duplicates. RNA-Seq based quantification of expression levels. So we looked at standard mapping pipelines. We did touch on novel isoforms, not in detail as we hoped to, but we got through a lot of it. We also looked at gene set enrichment analysis, and this is just the output here. You can see that the genes are annotated with gene ontology and gene descriptions, as well as you can get all the way to pathway enrichment and see which genes within the pathways are underexpressed or overexpressed, as well as we looked at some of the machine learning methods, analyzing expression data. We looked at unsupervised methods like PCA and clustering techniques, and then we looked at how to build a supervised analysis using such methods as SVM. And so again, to mention the data that we used data from 2 publications, cell line-based modeling of precision treatment of breast cancer, a project that is available on our educational site, as well as whole transcriptome profiling of PDX models as a tool to identify both tumor and stromal specific biomarkers. And you can find the link to this publication as well. And it's also a project that is available on our educational site that you can play around with. And finally, thank you for Dr. Vladimir Galatenko, who is the chief scientist at the Tauber Bioinformatics Research Center, and Julia Panov, who is the PhD student that uses the platform in some of our research projects. And again, special thank you to Dr. Javid Iqbal from UNMC, who helped organize the room and brought a lot of the people to the workshop. And we will be staying in touch by email. If you want to email Sahil, myself, or schedule a time, we plan to be there again and organizing another workshop. If you were not able to attend, or if somebody that you think should attend a workshop like this, please let them know. On the 20th, we will have another workshop at UNL in Lincoln. You can ask Sahil about that workshop. It will cover most of the same steps that we did today, but we will also touch on some new things. Again, thank you all for coming.
Thank you, Ilya.
Thank you, Sahil.